Hi, I'm Samantha Lewis with Cambridge Health Tech Institute, and I'm here at the 10th Annual BioIT World Expo with Khimani and Patrick Donovan of PerkinElmer. They have a poster here at BioIT World on structuring data in an ELN environment for biologics research, and I'm going to hand it over to them to explain it further.
Thank you. Anis here. The Ensemble for Biology is an informatics platform that PerkinElmer launched last week, and we are presenting that platform here at the BioIT World Show. And to go with that, we have a poster that my colleague Patrick here is going to talk about. Ensemble for Biology is an informatics platform that enables collaboration, paperless advantages to the using the ELN, as well as it integrates all of the data management, knowledge management into the drug discovery and development workflows, and enables scientists across various stages in the drug discovery process to collaborate across the various silos and departments within those stages. The platform represents a portfolio of products within informatics, which is the ELN, the Electronic Lab Notebook, for biology and chemistry from PerkinElmer Informatics, as well as the BioDraw and the ChemBioDraw products, and integration through our LIMS link at PerkinElmer of the instrumentation through the different workflows within that drug discovery and development process. Having said that, I'm going to pass it over to Patrick, who can walk you through the work that went into the poster and the highlights of the poster that enabled the workflow through Ensemble for Biology.
Thank you, Anis. So within this poster, what we're really explaining is how actually at a technical level and some of the choices we made in defining the architecture of the Ensemble for Biology suite. Within biologics research, typically the workflow is often carried out by specific groups that produce essentially data blocks. Here we have an image of that where a cell line group may be growing their cells, but they need to pass on the products to be purified and analyzed by subsequent groups. So really the trick is to analyze all this data together and to stitch it together. Because that's really ultimately how it's going to be useful. Now, in a traditional notebook, oftentimes the data that we actually capture is unstructured. So it may reside in Excel spreadsheets. It may reside in like a Word document or something like that. So it's actually very difficult to extract the data and put it together in a way that makes sense in the overall context of the research experiment. So if you think about it, there are many different groups. Each one of those groups has some bit of data that they're producing. And so actually putting it all into one piece can be very challenging when you're starting from an unstructured data format. So within our application, what we do is we actually take this data and make it such that you can define data blocks for each one of these areas in a research group. For example, your cell line development group may have certain products or other data that they generate, and each one of those can be attached to other groups' data within the application itself. Now, at a technical level, that's very tricky because databases it does require a certain amount of technical expertise to do that. However, what we've done is actually implement it such that you can manage all of these data blocks within the notebook itself. And what that allows you to do is put structure around your data, tie it all together, and make it accessible for the end users. And ultimately, that's going to have a lot more value to companies able to mine data out and extract it and analyze it later on. Thank you.
Okay, and again, that was PerkinElmer here at the BioIT World Expo.